we are studying about the topic inheritance and variation. Already we have studied meaning of word inheritance, that is transmission of characters from one generation to other, and the word variation means difference between the parent and the offspring. In the last lecture we have studied about mono-hybrid cross, which is in front of you once again. What is the meaning of mono-hybrid? Mono for one. So when two individuals, or we can say two parents, differing in one pair of characters. So here this is one of the parent, homozygous tall, and this is another one which is homozygous dwarf. So these are the two parents differing in one pair of character, that is tall and dwarf, are crossed together. We'll get F1 generation, which shows all tall individual, all tall plants which are heterozygous. And when these are crossed together, that means when the self-pollination of F1 hybrid takes place, it results in formation of F2 generation, which shows phenotypic ratio 3:1 and genotypic ratio as 1:2:1. So here one of the character of height is considered. But Mendel in his experiment have studied 7 pairs of characters. What are these 7 pairs of characters? We'll study that first and then we'll go forward. So we'll see that 7 pairs of characters now. So here these are the 7 pairs of contrasting characters which are studied by Mendel for his experiment. These 7 pairs of characters, first one is about seed shape. You can see in the diagram 2 types of seed shape are there. First one is round and the another one is wrinkled. In this round and wrinkled shape of the seed, round is a dominant character. This round one is dominant and wrinkled is recessive. We'll see the second character, that is seed color. 2 types of seed colors are seen, yellow and green. Yellow is dominant over the green. Then we'll see about pod shape. 2 types of pod shapes are there, inflated and constricted. This inflated is dominant one, constricted is recessive character. We'll see about the pod color. This green is dominant over the yellow color of the pod. About flower color, purple and white. Here purple is dominant over the white color of the flower. About flower location. 2 types of location are seen. One is axial. When the flower develops in the axil of the leaf, it is called as axillary or axial position. And when it develops at the tip of the stem, then it will become terminal position. So axial is a dominant character over terminal position of the flower. Last character is about plant size, that is tall and dwarf. Tall and short. Tall is dominant over this short plant. So these 7 characters were studied by the Mendel. Even though these 7 characters are there, these characters were studied individually one at a time or in combination of 2 or 3 characters at a time. Why Mendel was successful in his experiment? There are some reasons why Mendel was successful in his experiment. We will see the reasons for Mendel's success. So, these are the reasons for Mendel's success. First one, his experiments were carefully planned and involved large sample. That means a large number of seeds were sown and each character was studied very carefully and in a planned manner. Secondly, he carefully recorded the number of plants of each type and expressed his results in ratios. That means, as we have seen, phenotypic ratio 3:1, genotypic ratio is 1:2:1. So all this data, whatever is there, so all this data was carefully recorded for the particular number of plants showing a particular character, and these were expressed in ratios. As we have seen in pea plants, contrasting characters can be easily recognized. As we have seen just now a diagram showing contrasting characters. So these characters which are present like round and wrinkled seed, yellow and green seed color, like inflated and constricted pod shape, all these characters are seen very properly, and because of that we can identify the contrasting characters and their inheritance. Next to that, 7 different characters are controlled by single factor each. During Mendel's time, genes were not discovered and hence they were called factors. So for 7 different characters, 7 different factors were present, and these different characters were controlled by different factors which are present on different chromosomes. So it was easy to study their inheritance pattern. Next to that, the factors are located on separate chromosome and transmitted from one generation to other, as I said earlier. And he introduced the concept of dominance and recessiveness. Because of all these reasons, because of his careful experiments, hybridization technique, as he recorded the data mathematically and statistically, it was possible to give the results, and hence all these are the reasons for Mendel's success. Question of 2 marks can be asked on this. Okay, so in the topic we have started with the definitions of heredity and variation. 1 mark question can be asked. Give definition. After that, we have studied about the monohybrid cross, and in relation with monohybrid cross, we have studied about many of the genetic terminologies. I think all terms are clear for you, as well as monohybrid cross with its ratios is also clear to you. Now we are going to study about dihybrid cross. So dihybrid, meaning of word di for 2, hybrid for the mixing of characters. So we are going to study about dihybrid cross. What is that? So now dihybrid cross is there in front of you. What is the definition? We will see first. A cross between parents differing in 2 heritable traits is called as dihybrid cross. For example, cross of pure tall and rounded seed plant with dwarf wrinkled seed plant. Here in this diagram, in the slide, here 2 different characters like rounded seed and yellow color of the seed. These 2 characters are considered. So what is this cross? Cross between parents. This is one of the parent, this is second parents. Differing in what? Differing in 2 heritable trait. Which are these 2 heritable trait? This round yellow and wrinkled green. So these are the 2 heritable traits, or we can say differing in 2 pairs of contrasting characters. When these plants are crossed, it is called as dihybrid cross. So we are going to study about this cross now. This is the parental generation, and phenotype of the parental generation is one of the parent consists of round and yellow seeds. Here the genotype used for the round is capital R, and for yellow it is capital Y. Why this letters we have to use 2 times? Because genes are always present in pairs as they are situated on homologous chromosomes. So for rounded yellow, capital R, capital R, capital Y is the genotype. And for wrinkled green, the genotype is small r, small y, small y. This wrinkled, for the wrinkled seeds, genotype is small r, and for green color of the seed is for small y. So, parents differing in two pairs of contrasting characters, when these plants are crossed, it will become dihybrid cross. So, now we have genotype for round yellow and wrinkled green seeds. We'll go to the next step of gamete formation. So, during gamete formation, what happens? From this homozygous condition for the rounded seed, this R is come into the gamete, and from this pair of Y, one of the capital Y come into the gamete. That is why it happens so. It is because of reductional division. Half number of the chromosomes will enter into the gamete. Same is the condition in case of wrinkled green. That is small r from this set of R and small y from this set of Y will come into the gamete. Again, the number is reduced to half. So, this is the step of gamete formation. Now, these gametes fuse together in the process of fertilization and results in formation of F1 generation. So, this capital R with small r, capital Y with small y, this is the hybrid condition for round and yellow seeds. We have studied that in F1 generation we got heterozygous condition, and in heterozygous condition one is the capital gene is present, that means one of the dominant gene is present, and the gene which is dominant, it suppresses the effect of recessive gene, and that's why in F1 generation the plants will be getting with rounded and yellow color of the seed. So these are the hybrids for round and yellow color of the seed. In the next part, we have to study about gamete formation from this plant, from this F1 hybrid plant. So we'll see the next slide. How the gamete formation takes place. Now see, for gamete formation from F1 hybrid, this capital R, capital Y, small y, it is a F1 hybrid. Now we have to form the gamete. So what will happen? This capital R will combine with this capital Y. So the first type of gamete form is capital R with capital Y. Second type of gamete form is this small r will combine with this capital Y. So what we have seen? Capital R combined with this capital Y and small r combined with this capital Y. Now we'll see the next form. This capital R combined with this small y, and the next one will be small r combined with this small y. That means this is a condition where both dominant genes are present. So, how to form the gamete? We can start with one of the letter. This capital R combined with this capital Y, and capital R combined with this capital Y. So, capital R, capital Y, this is one of the gamete. Capital R with small y, this is second type of gamete. Then we are going to start with this second letter, that is small r, second gene, that means. So small r will combine with this capital Y, this is one of the gamete, as well as this small r combined with this small y, this is the fourth type of gamete. So in case of dihybrid condition, F1 generation shows the dihybrid condition and forms four types of gametes. So when these F1 individuals are selfed, when they are crossed with each other, what type of genotype and phenotype will getting? This is shown in the next chart. Now see, This is a dihybrid cross. Once again, parental generation with round yellow seeds, and second parent is with wrinkled and green seed. Here genotype capital R, capital R, capital Y, and here it is small r, small y and small y. So this is homozygous condition for the dominant characters, and this is homozygous condition for the recessive characters. Second step is gamete formation. So capital R with capital Y and here small r with small y. When these two gametes fuse together, results in formation of F1 generation with hybrid or heterozygous condition. That is capital R, capital Y, small y. Okay, so now these gametes are arranged here. These are self-crossed. As we have seen gamete formation in the previous slide. These gametes are arranged on both the sides of the Punnett square. So these 4 types of gametes we can consider for the male plant and 4 types of gametes from the female plants. So during this crossing, we'll consider these 2 gametes fused together. Results in formation of capital R and capital Y. That means complete homozygous dominant condition, and because of that these seeds will be round and yellow. This condition, this gamete when fused with this one, then there will be combination of capital R with small r and both dominant condition for this yellow color of the seed, and because of that phenotype will be round yellow. Next is this capital Y, capital R fused with capital R, small y. So here this is the homozygous condition for the rounded seed, heterozygous condition for the yellow color, and hence it will be round yellow. When this gamete fuse with this small r, small y, here the complete hybrid condition is formed, but the seed is rounded and yellow, because here this capital R, this gene is dominant over this small r, and capital Y is dominant over small y. So here it is rounded and yellow seeds are formed. In the same way, here crossed are made with the gametes on both the sides, and we have got total 16 type of combinations. In this 16 type of combination, if you are going to count, round yellow seeds will be getting this 4, 5, 6, 7, 8 and 9. That means 9 seeds are round and yellow. If you are going to see wrinkled yellow condition with this type of genotype, small r, capital Y, it is wrinkled for this and capital Y is for yellow. So this is wrinkled yellow condition. This is also for wrinkled yellow condition. Small r for wrinkled, capital Y, small y is hybrid. So we'll be getting 3 plants as wrinkled yellow, 1, 2 and 3. So 9 are round yellow, 3 are wrinkled yellow, these 3 are round and green, and only 1 is wrinkled green seed. So what will be the phenotypic ratio? Phenotypic ratio is here 9:3:3:1 in case of dihybrid plant. And what will be the genotypic ratio? Genotypic ratio will be 1:2:4:1:2:2:1. That means genotypic and phenotypic ratio is different. So remember that for the dihybrid cross we will be getting 16 different type of combination, and if you are going to sort it out, it will be 9:3:3:1 for phenotypic ratio. Okay, so this is all about dihybrid cross. After that, we have to study about Mendel's laws of inheritance. What are these laws of inheritance? See, as I said, Mendel proposed 3 basic postulates on the basis of which 3 laws were formulated. And these 3 laws were formulated by the scientists like Coren. Okay, so we are going to study about Mendelian laws of inheritance. We will study the first law of inheritance. First law of inheritance is law of dominance. We will see the law of dominance only. What is this law of dominance? See, when two homozygous individuals with one or more sets of contrasting characters are crossed, the allele that appear in F1 generation are called as dominant, and those do not appear in F1 are called as recessive. As we have seen, monohybrid and dihybrid crosses both. You have seen that. Once again, we'll see a cross. See, this is the dihybrid cross where round yellow seeds are crossed with wrinkled green seeds. The gamete formed are this type, and the F1 generation shows round yellow type of seeds. That means what? This round and yellow color of the seed are dominant over wrinkled and green color of the seed. So what we have seen, the character which appear in F1 generation are called as dominant, and those which are not able to express itself are called as recessive. So we can say that wrinkled and green color, these two are the recessive characters. We have seen in case of monohybrid cross also. In case of monohybrid cross, when tall plant is crossed with dwarf plant, in F1 generation we got only tall plants. So tallness is dominant over dwarfness, and we can say that this dwarfness is a recessive character which cannot express itself in F1 generation. So this is the law of first law. This first law of Mendel is the law of dominance. So this is the first law. Now we are going to see about law of segregation, which is also called as law of purity of gametes. So we have seen that during gamete formation, the alleles segregate or they separate from each other and enter in different gametes. So F1 hybrid, as we have seen, F1 hybrid like capital T, small t in case of monohybrid cross. And when they undergoes the formation of gametes, there is separation of gene capital T and small t takes place, and two different gametes are formed. So that is the law of segregation of gametes, or which is also called as law of purity of gametes. So the gametes formed are pure, and that they carry only one allele each, either dominant or recessive allele. Hence this law is also described as law of purity of gamete. Once again we'll see the monohybrid cross here. We have seen before. Again here monohybrid cross. What is this segregation of law of segregation? Here this capital T small t, it is the hybrid plant. When it undergoes the gamete formation, this gene capital T gets separate from small t. So these two types of gametes are formed. Even though they remain together for a longer period of time, they do not contaminate each other and enter into the separate gametes. As they do not contaminate each other, there is no blending, there is no mixing of the genes takes place. Hence we can say that they are pure, and hence this law of segregation is also called as law of purity of gametes. Okay, so remember law of dominance and law of purity of gametes. And now we are going to